Now let's discuss about bioassay. Okay, so what is bioassay? So bioassay is a experimental process by which uh, determination determination of concentration of biological active substance is performed by using biological biological indicator with reference standard. So here what we are dealing with is a process in which, as you can see here, it actually help to estimate. Okay, estimate, that means what we are saying, a determination of the concentration of biologically active substance. Okay, so here with the help of this process, okay, what we are doing, we are estimating the amount. Whose amount we are estimating here? We are estimating the amount of this biological active substance. Okay, that means the estimation of the amount of the biological active substance. Now in an experiment or in an experimental process, this biological active substance will be the unknown sample, as because here what we are doing, we are estimating the amount of that. Now just look into this definition. One is by using the biological indicator with the reference standard. Okay, so here the estimation of the amount of the biological active substance, that means the unknown sample, this has to be compared with the reference. Now this estimation of the amount of the biological active substance, okay, this we are doing by a unit quantity of the preparation, isn't it? So here you can see that within a bioassay, what we are doing, we are estimating the amount of the biologically active substance unit quantity of the preparations. Okay, that means we are determining the unit quantity of the preparation of that unknown sample. Here we are comparing with the standard, with the reference standard. Now while doing the reference standard, keep in mind that whatever the condition required need to be fulfilled. So here we need a rigid environmental condition, which is control. Okay, these conditions should be controlled. Within a control environmental or laboratory conditions, the whole process of the experiment has to be carried out. Now looking into this definition again, here we have mentioned the biological indicators. That means the we will be determining the concentration of the biological active substance or the unknown sample by using the biological indicator. Now what do you mean by this biological indicator? The biological indicators are the experimental animals. Okay, it are the experimental animals that will be used in order to perform the bioassay. The experimental animals, or it may be we can take that isolated tissue. We can also take the isolated tissue. Okay, we can isolated tissue or immune cells also. Okay, immune cells. Microorganism can be also taken in order to perform. So all these are nothing but they are all the biological indicators. Okay, these are all biological indicators. So at last, if we summarize what is actually bioassay, we will say that bioassay is actually an experimental procedure through which we will be able to determine the concentration of a biological active substance using the biological indicator with a reference standard.